The gut microbiota and the short-chain fatty acids that they produce can affect your energy absorption and your weight gain. Thus, the gut flora might have an impact on whether a person is fat or thin. Short-chain fatty acids are formed during bacterial fermentation of carbohydrates in the colon. The short-chain fatty acids cause low intestinal pH from the end of the small intestine to the beginning of the large intestine, and this prevents the overgrowth of bad bacteria like Clostridia. The short-chain fatty acids help to fix a leaky gut, strengthening the intestinal wall by increasing the secretion of mucin 2, and this then prevents lipopolysaccharides from crossing the barrier. Out of the total short-chain fatty acids present in the colon, 90 to 95% are acetate, propionate, and butyrate. In healthy people, it's around 60% acetate, 25% propionate, and 15% butyrate. Most of the short-chain fatty acids are absorbed in the colon, being exchanged with bicarbonate. The short-chain fatty acids are acidic, while bicarbonate is alkaline. There's some conflicting information on the short-chain fatty acids and weight. On the one hand, they increase caloric utilization, but on the other hand, they've been inversely associated with obesity. Overall, butyrate seems to be broadly protective against obesity. Propionate has mixed associations, and acetate is linked to weight gain. The fecal concentration of the short-chain fatty acids is 20% higher in obese individuals than in their lean counterparts. According to some researchers, this might reflect a compensatory protective mechanism against obesity, in which a greater amount is eliminated from the stool. This would prevent the increased accumulation of the short-chain fatty acids in the intestines, which can cause weight gain, mainly from the acetate. The short-chain fatty acids, like butyrate and propionate, increase the formation of the gut hormones glucagon-like peptide 1, or GLP-1, and polypeptide YY, and these reduce the food intake by decreasing appetite. The butyrate reduces food intake, and it's been linked to weight loss. Researchers are also investigating butyrate's potential to combat autoimmunity, cancer, and psychological disorders. Butyrate, which is primarily produced by Firmicutes bacteria in the gut, appears to affect gene expression in the brain. Butyrate is primarily used by the colon cells as a major source of energy. The butyrate production is affected by the quantity of bacteria that produce the butyrate and by the pH of the large intestine. Butyrate-producing bacteria seem to thrive in a more acidic environment, whereas the acetate and propionate bacteria seem to thrive in a more alkaline environment. In the mitochondria of colon cells, 70 to 90% of the butyrate is oxidized into acetyl-CoA, and this is subsequently processed through the tricarboxylic acid cycle to generate large quantities of cellular energy in the form of ATP. Propionate also reaches the liver via the portal circulation, where it's used to produce glucose. The propionate has been observed to have effects that promote or suppress obesity, but overall it seems to be mostly protective against obesity. Of all the three short-chain fatty acids, the acetate seems to cause the most weight gain. However, it also has some properties that are believed to protect against obesity. According to some researchers, it might act against weight gain in the hypothalamus by increasing GABA. Human studies have shown that lactulose, which is metabolized by the microbiota to produce high amounts of acetate, when administered to the diet of 6 volunteers for 2 weeks, showed a significant increase in both the total and LDL cholesterol, the ApoB, and blood levels of acetate. Obese animals and humans have a lower microbiome diversity. Methane-producing archaea have been found to be present in greater abundance in obese mice and humans when compared to lean subjects. Colonization with M. smithi and B. tetamicron led to the fermentation of dietary fructans to acetate, and this resulted in a significant increase in fat creation. M. smithi is found in 70% of human beings. It generates methane. It's been found to enhance the fermentation of polysaccharides and other carbohydrates by removing hydrogen atoms, leading to greater production of short-chain fatty acids and hence their increased absorption. These short-chain fatty acids function as an extra source of energy, and this can contribute to the weight gain and the obesity. The number of hydrogen-producing strains were at a higher level in obese individuals compared to lean subjects, with those after gastric bypass. The investigators hypothesized that hydrogen was reduced, and this allowed a greater production of short-chain fatty acids, and this led to more energy that's then absorbed across the intestines. Proteilaceae is a source of lipopolysaccharides, and this causes inflammation. C. ramosum in the mucosa of the small intestine of mice increased body fat as a result of the increase in the intestinal absorption of glucose and lipids. The intestines of obese humans and mice have been found to contain low relative proportions of Bacteroides vulgatus, high Firmicutes, and high Clostridium ramosum. High lactobacillus species, which was positively correlated with high C-reactive protein levels. High Lactobacillus reuteri and high Staphylococcus species, which was linked to the increased caloric intake in children. Oscillobacter and Clostridium cluster, which was found in obese prone mice and was totally absent from their obese resistant counterparts. Clostridium leptum, which was found to be associated with both obesity and weight loss. F. praunitzii, which is linked to the alteration in the inflammatory state and diabetes. Resistant starches and dietary carbohydrates that humans can't digest on its own need certain gut bacteria to ferment them into the short-chain fatty acids, primarily the butyrate. Some researchers have argued that eating a diet rich in the resistant starch could promote the growth of the butyrate-producing bacteria. Resistant starch can be bought in a powdered form, for example as high maize, or obtained from dietary sources like some whole grains, green bananas, cooked and cooled starchy foods like boiled potatoes, cooked legumes, and some tubers like yam. To book a consultation or to learn about herbs, supplements, and natural treatment plans, check out my website.